I'm very excited to be here. I enjoy collaborating with entomologists. I'm definitely very naive when it comes to insect biology, so I appreciate when I interact with and collaborate with entomologists, and I'm very excited and humbled to be part of this meeting. And I'm wearing the only shirt I have that has an insect theme. So I'm excited about being here. As you heard, I'm an associate professor in EEB, Ecology and Evolutionary Biology, but I also direct the Spatial Analysis Lab in NIMBioS, and I work there with Eric Carr, who organized the virtual part of this meeting, and we try to build, in the Spatial Analysis Lab, we try to build a cross-disciplinary environment. We help people with spatial data analysis, remote sensing, and we also help researchers with proposal submission if they require, or their proposal can be enhanced by contribution, a spatial analysis contribution. So I'm just putting that out there. If anyone is interested, please feel free to contact us about the Spatial Analysis Lab. Okay, so generally I started with a disclaimer that I'm not an entomologist and I'm humbled to be here. So generally my research interests fall or can be grouped under this big umbrella of determinants of species distributions. And within this umbrella, I'm interested in methods that can be used to study species distributions. And I am interested in the methods in terms of applications to macroecology, conservation biology, and sometimes disease ecology, again, with the collaborators, because I'm not a disease ecologist. And I provided a little word cloud on the right side of the screen, just to briefly mention my broad research interests. So why am I interested, and many other people, why are we interested in species distributions? Well, first of all, we all understand that these are spatially explicit characteristics of species. So we do need both ecological knowledge and spatial knowledge or spatial information to study these to study species distributions. And I'm showing just a visual example of what I mean by spatially explicit distribution and the ecological background of that spatial distribution. So we have species of conservation concern from South America, Dasypus species that has a known range, an IUCN map in this outline in green. And then we can, in ecological space or in environmental space, in this case, annual precipitation and annual mean temperature, we can graph the ecological, the broad ecological or climatic, in this case, two-dimensional climatic requirements of this species. So within that big range, these red dots on the graph represent the red dots, the known individuals of this rare species. So by combining spatial and ecological knowledge or information available for a species, we can further understand limitations, biogeography for the species or for species in general, and that can have various applications, as I mentioned. So for today, my plan is to provide a very brief introduction into ecological niche modeling, or at least I have, I think, 3 slides. I don't know if I will be brief, but I'll try. So I would like to provide a brief overview of ecological niche modeling, this methodology, and then illustrate this method, this research method with 2 case studies, and focusing on forecasting potential distributions of species under climate change scenarios. So the 2 case studies of forecasted potential distributions are Great Plains pollinators of conservation concern, a study I did a few years ago, and then a recent study of pollinators, buzz-pollinators that are important for tomato crop. What I would like to do is encourage questions throughout at the end of each of these three sections, because I know it's really hard to focus on Zoom. It's generally hard to focus on a seminar that's 50 minutes long, and I think it's much harder, much more difficult based on 6 months of Zoom experience or Zoom hell, whatever, however you want to call it. We know it's really hard to focus for 50 minutes on a Zoom presentation. So what I will try to do is encourage questions after each section. So I'll have a slide that prompts you to ask questions if you have any. And I guess Greg or Carla or Eric will be monitoring the question and answer feature of Zoom. Okay, so let's start with this hopefully brief introduction into ecological niche modeling. So that's my first section. I wanted to be clear about what I can do and what generally we can do with this approach, this ecological niche modeling approach. So in order to get a sense of what we can do with this methodology, I thought I would make you think about the geographic distribution of species, where species are found in geography. So if we work at small geographic extent versus large geographic extent, and by that I mean maybe Smoky Mountains National Park in terms of small geographic extent, even though it's a big park, but in my macroecological mind, that is a small geographic extent. And then at the other end of the spectrum of this distribution or geographic space is this large geographic extent. Think about an entire continent, or think about, I don't know, Eastern United States. To me, that those are large geographic extent. So if we think about what is the extent, the geographic extent of my question, then we can start thinking about what kind of factors influence the presence of the species at small geographic extent versus at large geographic extent. So at small geographic extent, we have plenty of evidence that species interactions, biotic interactions, are very important in determining species presence in a community in a small geographic extent. So if our question and our study is at small geographic extent, then we have to really pay attention to the role that the species plays in that region, that small geographic region, and the interactions that the species has with the environment. And that is because the presence of the species has an impact in that, in terms of resources available, in terms of competition, so on and so forth. So this is maybe at community level that we are thinking about species presence at these small geographic extents. So it is clearer then that we need to focus on relationships between species, not just abiotic conditions that are suitable for a species at that extent, and probably the kind of modeling, or likely, based on available literature, the likely modeling approach for modeling species presence at these small extents is a dynamic modeling approach. Now, if we, meaning population dynamics, meaning consumers impacting availability of resources, so on and so forth. Now, if we switch to the other end of the spectrum of this geographic extent spectrum to large geographic extent, that's where the dominant factors of species distributions become the more important factors become the abiotic ones, not the biotic factors. Generally speaking, there are new and recent studies that show that depending on the species, the biotic interactions might actually be quite important, or as important as abiotic factors at these large extents. But generally, for many species, for many examples we have in the literature, at these large geographic extents, continental, Eastern North America, a big geographic region, we can rely on abiotic factors, on the requirements that species have in order to survive in a region, as the main determinants of species distribution. So because we assume and we have evidence that species requirements are stronger than species interactions at these large extents, we are using this ecological niche modeling approach that relies on empirical modeling or correlative modeling. And I will explain, this is the area that I'm active and I understand better than the dynamic modeling. Okay, so specifically with ecological niche modeling, what we are doing is we are using correlative algorithms to estimate species distributions based on their environmental requirements. So because the approach is making use of known presences and environmental conditions, the data requirements or the data needs for ecological niche modeling, at least at a first approximation, are quite clear or simple. So we have species records, locations with latitude and longitude, so spatially informed locations of observations or collections of a species of interest. And then we have a set of predictor variables, these environmental conditions, in this case, in this example, climatic conditions, so digital maps of precipitation of the driest month, precipitation of temperature, seasonality, so on and so forth. So we have these two types of data inputs that allow us to calibrate a model that estimates the requirements of the species, the requirements that allow the species to occupy, to be present at these locations. So with ecological niche modeling, with these data, presence data and environmental data, we try to estimate the abiotic conditions or estimate the niche that is suitable for that species. And then once we have that model calibrated, we can project that model on the same, or there could be another region, but in this case, same geographic region, the Great Plains, and we get the potential distribution of the species, meaning the locations in geography that are environmentally suitable or similar to the known presences of the species based on this model calibration. So that's the basic data input and data output from these with this ecological niche modeling approach. Now, for today's the studies I'm showing today, the two studies, we used an algorithm called Maxend, which stands for maximum entropy. So Maxend uses environmental data as predictor variables and presence data of a species as input, but it also samples the background or the rest of the study area. It samples random locations that are not associated with species presence. It samples the environmental conditions at those non-presence locations. So that's the background of the locations for which we don't have presences of the species in our study area. This algorithm requires, if you are familiar with, for example, presence absence, like a logistic modeling approach, presence absence or other, and I mentioned logistic modeling, but in general, presence absence modeling approaches require presence data and obviously absence data. Now Maxend, and usually that has to be, the presence and absence data sets have to be balanced. Maxend works different in a different way, meaning that it requires a large sample of the background, a large sample of the non-presences, in order to be able to find those environmental differences between known presences and the rest of the study area. So this is an illustration from a paper I forgot to cite. I think it is Elith et al., if I remember well. So we have the predicted variables or covariates, temperature, precipitation, so on and so forth, many variables. Then we have presence records, known presences, and then we have the background samples. Like I said, a large number of background sample. And then for each of these covariates, the algorithm can figure out what are what is the probability density, what is the likelihood that a certain combination, in this case temperature and precipitation, is associated with presence. So in this case, we see a peak over here versus the rest of the locations or the pixels at the study in our study area. So it's contrasting combinations of environmental variables at presences, contrasting that to background samples. Okay, this is where you can ask questions, or you can get a sip of coffee or pastry or anything like that. So if anyone has questions, I'll wait for 1 minute.
Mona, I don't see any in the Q&A, but I have one to throw at you. This Maxend algorithm, is that something that is sort of a standard package program, or are these built by you or modified by you?
Yeah, I'm just a user. I don't code. I just take whatever smarter people design in terms of modeling. So specifically Maxend. Maxend is an algorithm that was published in 2006, I think. And when it was first really by researchers from City University of New York, and I think a researcher from, doesn't matter, but the main researcher was at City University of New York, Rob Anderson. And so when it was first proposed as a modeling, ecological niche modeling algorithm, it was code programmed in Java. So the programmers created a GUI interface, like a nice interface with menus that we can, options, parameters that we can use. So so it's very easy to use. It's free to download. If you Google Maxend, you will get to the repository for the Java application, which is at American Museum in New York. So AMNH, if you Google Maxend AMNH, you will get to the download page. But so that was the start. That was 2006. Maybe in 2009 or so, the first R package was released for species distribution modeling, which is another way of saying ecological niche modeling, more or less. We're not going to get into the debate of terminology. So this R package has several algorithms, including Maxend, that you can run under R. And so that was 2009, and it's 2020 now. There are several R packages that have fine-tuned the way you run models under R. So there are many options, but the Dismo package, ENM eval, and KU ENM are the most common ones, I think, that are used under R. So yeah, plenty. There's the GUI interface that I use, very comfortable with it, and then there are plenty more packages, at least 3 that I'm aware of under R.
Thank you. We had a bunch of questions come in as you were talking, so I'm going to go through them. The first question is, does this type of modeling work for aquatic species as well as terrestrial species? And will it work for endemics that occur only in one watershed?
Very good question. So I'm a terrestrial ecologist by training. So after I finished my PhD at University of Kansas, I went to University of Wisconsin in the Center for Limnology. And it was a massive change in thinking of ecological niche modeling, because I had to create environmental data sets for aquatic species. And it's really hard. I will say this, it is really hard, but there are lots and lots of, so there are entire labs and research groups that work on modeling the niches and potential distributions of aquatic species. The challenge is to get those environmental parameters at maybe the stream segment scale that then represents maybe, I don't know, the temperature at the watershed level, which is your study area. So yes, it is possible, but the environmental data require a bit more harder work than what we do in terrestrial ecology, which is we have lots of already generated climatic variables, land use variables, so on and so forth.
Thank you. Okay, the next question is, can you elaborate on what the background sample is? You mentioned that it needed to be larger, so can you explain kind of what that is?
Yeah, so if we have a small background sample, chances are small, meaning maybe similar to the size of presence data. What happens is, by chance, this random grabbing or random sampling of pixels of cells that are not associated with presences, this random subsampling might not have enough environmental heterogeneity, might not be that different environmentally from our presences. So then the model is not able to, doesn't see any different peaks in the density of records in multidimensional space. So it's not able to distinguish environmental conditions associated with presence and the rest of the study area. So that's why the more samples we provide to the algorithm, meaning either the study area is large, so that we have lots and lots of locations, lots and lots of pixels in GIS terminology, or cells, or we have a small region, geographic region, but we have high resolution, so the pixel size is very small, so then we have more than 10,000 pixels. That's the at least the default limit, or the default requirement for Maxend modeling algorithm. So the reason for a large sample size, that a large number of locations that are not associated with presence, the reason for that is, or the need for that is, in order to be able to environmentally separate presence from the background. So.
Okay, thank you. Next question is, I was wondering about the potential of this to predict future range of imported fire ants or other insects. Is that something you're going to talk about today?
I'm gonna show you two examples of future predictions. Well, we shouldn't call them predictions. They're not predictions. Forecasts or estimates of potential distributions, and they can be for invasive species. They can be for species of conservation concern, which I have an example here, or important species like pollinators. Ecological niche modeling is heavily used for estimating potential distributions of invasive species. Really, lots and lots and lots of papers, and it's a very interesting field, because from basic ecology, it has interesting challenges. If a species has been recently introduced into a new area, let's say it's I don't know, European species. I'm from Romania. I invaded US. So if it's a European species that has been, let's say, accidentally introduced into US, then we will use the native range, the European known presences, to calibrate a model and then project the model on US, the same environmental variables, the same, let's say, climatic conditions, and we get a potential distribution map. But then the question is, how can I test this map if the introduction just happened? So there are interesting questions about equilibrium, species equilibrium with the environment, dispersal. So yeah, it's a whole interesting field, the invasion biology field that uses niche modeling. But yeah, it's big field.
Thank you. The next question is, how can habitat fragmentation be factored into ecological niche modeling?
Yeah, it's a hard one. So usually habitat fragmentation, so any alteration of the niche space of the species has to be taken into account after ecological niche modeling, because we don't have enough instances of a species surviving in fragmented habitat that we can include as a predictive variable in the model. So usually what we do is we generate an abiotic suitability map, and then on top of that we add fragmentation or land use change, and we refine the model output that way.
Okay. What is your process for determining which environmental parameters you will use in the model?
Yeah, so that is a problem. Just like decide what will be the extent of your study area. So we debate a lot how we should select environmental variables, but at the end of the day, we rely on what is available. So imagine you are tasked with estimating the potential distribution of an invasive species or a species of conservation concern. You will not yourself generate those environmental maps, those temperature maps and precipitation maps or solar radiation. So you'll have to go out meaning on the internet, and find these variables, these GIS rasters that already exist that climatology center or USGS have already generated for us. So we are limited in terms of variables we are using. We are limited to what is available already, what has been generated by other research groups. And then what we can do is we can decide of what is available, what makes sense for the biology of the species, what makes sense for the question that I'm trying to answer. So you select the variables. Of the variables available, you select the ones that are more likely to be influencing the distribution of the species, or important for the biology or natural history of the species.
Okay, last question. If you have a species and environmental data, just like you showed for Maxend, but also with absence data, so I'm assuming that means sampling and you know it's not there, what package would you typically use for that type of analysis?
Yeah, so the R packages I mentioned, Dismo, ENM eval, not KU ENM, but Dismo has, I can't remember if it's 15 different algorithms, and the majority of algorithms are actually presence absence. So Maxend is one of the few algorithms that works with presence and not presence or background. There is an old one, GARP, that works with presence only. Then there is an envelope approach, BioClim, that works with presences, but everything else, so the other, like the alphabet soup of artificial neural networks, so ANN, GLM, generalized linear models, generalized additive models, GAM, support vector machine, boosted regression trees, all those algorithms are based on availability of absence data. They are presence absence. They are designed for presence absence situations. So if you have robust absence data, then you have much more, many more options in terms of modeling algorithms.
That's all for questions for this section, so I guess you can move on.
Okay. Okay, so the first case study I wanted to show you is a project that my former PhD student Cassandra Walker did. And she graduated, and she's a wildlife biologist with US Fish and Wildlife. And coincidentally, this project, when she was a graduate student, this project was funded by US Fish and Wildlife, had nothing to do with her current job. Okay, so what we had here, our goal was to create a list, well, not create a list, go through lists of conservation, species of conservation concern, pollinators of conservation concern in the Great Plains, and then estimate the potential climate change effect on the distribution of these species. I think originally we had over 30 species from various state lists of state level lists of species of conservation concern. But we ended up with these 14 species because only for these 14 species we were able to find occurrences, presence records that we could run models. And we had different taxonomic groups represented, but also sample sizes were very different. So we had the smallest sample size, 11 known records, versus the largest sample size, over 9,000 presence records. And we also had species that were narrowly endemic, and then we had species that were found throughout North America. So we ran the models at the extent of these, the known extent of these species, but then we focused on the distribution in the plains. So very briefly, we calibrated these Maxend models with climatic variables from 1960 to 2000. So we call these the baseline climate or historical climate. And then we tested the performance of the models with two metrics, AUC and omission error. And so what I quickly wanted to show you was for these species, the 14 species of pollinators of conservation concern we had variation in terms of model performance. So in red, you have one of the metric called omission error. And you see that for several species, well, 3-4 species, the omission error was slightly higher than what we would like, which is 0.2, 20%. And then for the other metric, AUC, we see that the AUC metric actually was pretty good, so higher than our limit of 0.7, which in this case it's a minimum limit of 0.7. So for most species, the AUC was higher than what we wanted, so that's good. And for a couple of species, it was not. And you see that the omission error was also, in this case, not what we wanted. And in this case, like I said, omission error, we want the measured omission error to be below that red line. So most of the species performed well. We had 3 species for which the models were still acceptable, but in one case, in this case, not quite acceptable. So, and it depends on the sample size in general. Okay, so we were comfortable with the model performance. So what we did is then we projected these models on future climate variables. So where are these future climate variables coming? Well, they are coming from models, general circulation models. So these are models built by climatologists that are forecasting changes in temperature and precipitation based on very complex models that take into account atmosphere, ocean, land interactions, and emission scenarios, greenhouse gases that are forecasted to be in the atmosphere in 2050s and 2080s. And by 2050s, we actually mean an average of 30 years. So from 2040 to 2060, And for 2080s, it's from 2070 to 2090, if I remember well. So we have our baseline understanding of the climatic suitability for the species. We have that niche model, Maxend model, and then we project that model on the same climatic variables, but these variables are now representing future conditions for 2050 and 2080 under different global circulation models and emission scenarios. So for this study, we used three global circulation models that are predicting low change, moderate change, and then high change in precipitation and temperature. And for each of these global circulation models, we considered the temperature and precipitation under two greenhouse gas emission scenarios, the moderate RCP 4.5 and then the high RCP 8.5. So the moderate emission scenario assumes that the world, the society as a whole, will implement changes and will work towards reducing emissions from where we currently are, versus the high emission scenario assumes that the world as a whole, the society as a whole, doesn't do much, and we continue the output and the growth that we have currently. So you can think of RCP 8.5 as, business as usual. We don't change anything. Where are we going to be in towards the end of this century? So this is not something that I do at all. Again, we just download these climatic variables that are modeled with these global circulation models under different emission scenarios. Okay, so here's an example for one species. Here's the projected geographic distribution, potential distribution under current or from 1960 to 2000 under baseline climatic conditions. Then this is the future potential distribution of the species under that 4.5 moderate emission scenario. And then this is under the lowest global circulation model, but using the moderate emission scenario. And then this is what the potential distribution will look like in the future under the high global circulation model, high change, the same emission scenario 4.5. So we maintain the emission scenario the same, we compare within an emission scenario, moderate emission scenario, but we compare a global circulation model that predicts change or forecasts moderate, low change, in this case in the middle, low change versus high change in temperature and precipitation. So you see that for the low GCM, global circulation model, there is a range expansion, and then there is a range contraction in red. So this is what is lost, and in blue, this is what the species is, what the area that will be climatically suitable under these under this global circulation model. Now, if we look at the high change global circulation model, the range expansion is smaller, and the range contraction is smaller, but the no change is actually higher than in the lowest GCM. So it's slightly unexpected. So we did this for the 14 species. So what I'm going to show you is several graphs, and I'll try to see. I hope that you can see the patterns in these graphs. So just to get you oriented, this is a bamboo species, and what the graph is showing is area of the species, potential distribution of the species under the moderate emission scenario, so RCP 4.5, and then under the three different global circulation models: the low change, the medium change, and the high change, high temperature and precipitation change. So what we see is that the high GCM shows a higher range contraction than the low GCM. And so no range change is highest with the under the forecast from the low change in the global low change global circulation model. So this is for RCP 4.5. Now, if we look at RCP 8.5, so the high emission scenario, what we see is an increase in the range contraction, a decrease in no range change, marked, not significant, marked decrease in no range change. So basically, and a decrease in the range expansion. So basically, this species is projected to experience higher range contraction under the higher emission scenario. And based on all these, the three global circulation models. So I will show you graphs that illustrate these differences between global circulation model, low, medium, high, and emission scenario, 4.5 moderate and 8.5, which is high emission scenario. So here are the bumblebees. And just quickly looking at the graphs, I hope you notice that the top of the far left and far right have higher range contractions regardless of, or have higher range contractions, not regardless, especially in the 8.5 emission scenario, whereas the middle two species over here are not projected to experience such high range contraction. So you see that the white bars, hopefully you can see that the no range change bars are higher. So a larger area of no change, the area of no change will be larger than the area of range contraction, and same for range expansion. So 4 different bumblebee species, 2 that are more similar in terms of projected patterns, 2 that are less similar. Here are the skippers. Again, we see a variation. In this case, we see large range expansion for a couple of species, 3 species, and then we see large range contraction for 2 other species. We do the same for butterflies. 2 examples, quite opposite projected patterns. And then here are the moths. Again, we see variation in terms of range expansion versus range contraction. So in other words, this is to say, the potential effect of climate change will be very different, will be species specific. At least these models project species specific effects of climate change, and also project illustrate the variation of these projections depending on which climate change, which global circulation model we use, and which emission scenario we use. So very different results. Now, if we try to summarize all this into just species richness, so number of species, this map over here shows the current species richness, so under estimated species richness under current climatic conditions, and by current, remember, I mean 1960s to 2000. And then these are the projected species richness under a low change global circulation model and a moderate emission scenario. And over here in the lower right corner, we have the high change emissions, high change global circulation model and high emission scenario. So if we just compare the top left with the lower right corner, we see that the number of species, the dark blue represents the highest number of species, the species richness, highest species richness. The number of species overall is projected to decline in the Great Plains under the high global circulation model change of temperature and precipitation and the high emission scenario. It's not So if we compare this less dramatic change, so low GCM and moderate emission scenario, if we compare this top left corner or map to the current estimated potential species richness, the differences are not that big. So we see maybe we see in the northern Great Plains actually an increase. So we have more green over here than in current times. But number of species doesn't account for shifts. So it might be different species or a reassembly of communities of pollinators, and in this case it's just pollinators of conservation concern. Okay, so what I want, before we get too excited or too worried, what I want to point out is that these are scenarios, they are not predictions. So we have all kinds of global circulation models outputs. We have at least 4 emission scenarios. We have to consider all that variation and those uncertainties. So keep in mind, these are scenarios of future potential distributions, they are not predictions. And also keep in mind that I did not include dispersal, we did not include dispersal, and we did not include biotic interactions here. Questions or coffee break for whoever needs to stretch legs.
We can wait a second because last time it took a little while for people to ask questions.
I'll be fast with the last one.
I just have a quick question. Is there an optimal, number of specimen location hits that these models work under, or does it depend on the range size?
Yeah, it's a good question. It does depend on the range size. If the geographic range is relatively homogeneous, then a low sample size, low meaning 15, 20 presences, will actually work. It does depend on the species, and the second example I have, we had sample size, most of our sample sizes were over 50, we had 70, and we had some models that didn't perform well. So it is a combination of how heterogeneous the environment is, and are the presence points capturing that heterogeneity, or are they too heterogeneous for the niche modeling, ecological niche modeling algorithm to make sense of that, to find the maximum entropy, the bounding surfaces, environmental surfaces based on those presences.
Thank you. I don't see any more questions, so if you want to move on to the second one, that'd be great.
Yes. Okay. So the third case study showcases Greg Wiggins, my colleague from NIMBioS, and Luis Carrasco, who was a postdoc just until, I don't know, 3 weeks ago, 4 weeks ago. Luis was a postdoc at NIMBioS and in my department, and he is now in Japan. He has another postdoc fellowship at Tokyo University. And I wanted to highlight, so this project was part of the NIMBioS summer research experience for undergraduate students. And we had an amazing group of undergraduate students. So we had Abby, Ellie, and Brendan. They are mathematicians, and they jumped into this pool of pollination biology, and they did just amazing. They were amazing undergraduate students, and I'm very grateful that NIMBioS has these, or had, this year was the last year, had these summer research experiences. They attracted amazing, very capable undergraduate students. So it has been true pleasure and learning experience. So I wanted to highlight, and so Luis and Greg did most of the work for this project, and I get to talk about it. And the undergraduate students did a lot of the data analysis, the data crunching. Okay, so what we had here was a subset of buzz-pollinators for tomato crops. We had an initial list of maybe 24 species, but we either didn't have enough occurrences, we couldn't find enough presence points, or the information about whether the species was really truly pollinating tomatoes was not that clear, so we trimmed the initial list. Now here, see the sample sizes were smaller than the case of the Great Plains pollinators. 5 to 243 presence records for across these 15 species. We did the same approach, Maxend, calibrated, measured model performance, and I'm pointing out 2 species that had higher omission error. The blue arrows are illustrating or are indicating 2 species for which we had higher omission error than what we wanted. We would have wanted 0.25, so 25 at maximum of testing occurrences being omitted. So we had higher omission error for a couple of species. The AUC was above 7 for all species, so that was good. Okay, so then we did the same thing, projected based on this validation of the models, model performance. We projected the models to future climatic conditions. We used the same 3 global circulation models that predict low or forecast low, medium, and high changes, respectively, of precipitation and temperature. But we also added an ensemble GCM that is an average of 15 different GCFs. I'm not going to present that specifically, but I just wanted to plant that seed in your mind that there are ensemble GCMs that crunch all kinds of different GCMs into one. And then we use the same 2 greenhouse gas emission scenarios, and we have the same 2 time periods, but I'm going to show you just 2050 like I did in the previous case study, and I don't think I mentioned that. Okay, so here's an example of maps that we get out of, well, processed maps. These are processed already, maps that we get out of Maxend. So we can, in this graphic, we are showing the area loss on the x-axis from current to future. And then on the y-axis, we show the spatial shift, the distributional shift of the potential distribution from current to future. And by future, I'm showing just one GCM, the moderate GCM that projects moderate change in temperature and precipitation. I'm showing you the emission scenario, the moderate emission scenario, and 2050, the time frame, 2050s. So where you see yellow, that is projected loss of potential suitability, climatic suitability. The dark blue is potential gain of climatic suitability. So we have situations where we have low area loss. So area-wise, the current and future are comparable, but there is a spatial shift, a distributional shift. So we have bombus impatiens, generally comparable current to future potential distribution, but shifting northwards. Then we have, at the other end of the spectrum, we have high area loss and low shift. So that basically means that the climatic conditions for this species are projected to be unsuitable in much of the current distribution of the species. So this is no distributional shift, just loss. And then we have in this corner, the up right corner, we have high area loss and also high distributional shift. So you see a lot of yellow here, and you also see blue regions, meaning that that's where the shift occurs, the distributional shift. So this done for many species, for 14 species. I'm running out of time. Now, if we crunch all the variation into one graph for area loss, distributional area loss for just 2050, this is what it looks like. So we have on the x-axis, we have species, the species that we work with, the 14 species. And then these box plots show the variation across the global circulation models, the 3 global circulation models that we used, plus the ensemble that is an average of 15 global circulation models, and then the 2 emission scenarios. So in a nutshell, what we are trying to show with this graph is that for some species, so for example, this species, there is a high variation depending what GCM and what RCP you're using, the projected distributional loss will be quite different. Then we have some species over here for which the projected distributional loss is quite comparable across these GCMs and RCPs. So basically that's what we are trying to show. It matters what GCM we are using, and for some species, it matters more than for others. Now, we also did, in this case, we also we calculated species richness, current, and then future. And we calculated the change in species richness from current to future. And again, I'm showing you just, in this case, I'm showing you just one GCM, and to compare the 2 emission scenarios. So for buzz pollinators of tomatoes, what we see is an increase in number of species northwards and a decrease in number of species. So in red, a loss in number in species in the central part and eastern part of United States. Both emission scenarios show relatively similar patterns with some local variation. Okay, so then linking these projections of changes in species richness of buzz pollinators, linking that to where crops are grown currently in United States. So on the left side, you have the average change in number of species of pollinators of tomatoes. And on the right side, you have the, at the county level, proportion of crop area that is represented by tomatoes. So in the right-hand map shows areas that are important for tomato production, and the left side shows where what counties will have an average loss or gain in species. So green shows gain in species of pollinators, and red, pinkish, shows loss in number of pollinators. So just visually, we see that there's some concerning overlap between the 2. But if we combine those 2 maps into one map, now what we are looking at are purple regions that have high tomato crop area, or proportion, I should say. Proportion of crop area is high, but it also corresponds to loss or change in number of species of pollinators. So purple is not a good situation. Red is also relatively bad because it means those areas are projected to lose buzz pollinators that pollinate tomatoes. The blue areas are good because that's where we have, again, high proportion of tomato crops linked to no change or gain in number of pollinators of tomatoes. So where we see blue, it's pretty good. Where we see red to purple, it's pretty concerning. Again, based on a moderate GCM, and this is a 2050 moderate GCM and a moderate emission scenario. Okay, so conclusions, because I am really out of, I have 1 minute left. We forecasted the potential distributional shifts of several species under several global circulation models and 2 emission scenarios. What we observed was regional variation in species richness, change in species richness. And we identified regions that could possibly be, in which tomato production could be possibly affected by these regional shifts in buzz pollinator richness. So one question I want to leave you with before I stop talking is, I want you to think, I would like you to think, to think what are the possible uses, but also misuses and limitations of these estimates of potential distributions under climate change scenarios that are based on niche models and global circulation models and emission scenarios. So I leave you with that thought. But if you have any questions, I first thank you for your attention. And if you have any questions, I'll be happy to answer if we have any time left.
Thank you for sharing with us today. If anyone has any additional questions, please put them in the Q&A box. Here we have a question for you. Given the dramatic effect of climate change could have on native bee populations, have you seen any evidence that commercial beekeeping in these study ranges could produce any measurable effect, either positive or negative, to wild bee species when the climate change effect could be so great?
Yeah, I mean that's a management, that's a mitigation, I guess, strategy. With the tomatoes, the concern is that these are buzz-pollinated, so you need to have a buzz-pollinator, not the honeybee. You need to have a buzz-pollinator that is commercially, and there is one species, I can't remember which one, that is commercially available, but it's hard. And I'm sure there are models out there that estimate that the climate change effects on commercial species like the honeybee. It's a mitigation, it's a possible mitigation solution. Oh, in our paper, so this paper, I forgot to mention, this paper is now, the second case study is in press in Ecological Applications, and Luis Carrasco is the first author if you want to look for it. It's not out yet. We do talk about other mitigation possibilities in terms of habitat restoration, creating some sort of like stepping stones for dispersal of pollinators.
Thank you. I don't see any additional questions, but just a final question is, if there are people out there who wanted to collaborate with NIMBioS or you or Greg, what is that process, and what kind of interests would spark collaboration there?
Definitely the easiest, I get emails, I answer emails. The easiest is to email Greg. Is the brain behind the second study. So like I said, I'm an ecologist and I collaborate with entomologists. So, but generally for Spatial Analysis Lab, you can contact Eric, you can contact me for questions related to potential distributions of pollinators. By all means, contact Greg, and I'm sure all three of us are happy to help and answer questions or collaborate.
Hey, Mona, related to collaboration, I don't know if you elaborated on how much the spatial analysis has as far as like remote sensing equipment. That wasn't really covered in this lecture, but that's another whole aspect of spatial analysis, is your drone, hyperspectral drones, or other types of imaging that might help people model stuff. Yeah,
I had two pictures there, but I didn't mention what they were. Yes, I had a terrestrial LiDAR scanner in my second slide and the drone. And so yeah, Eric is the pilot for the drone. If you have any questions about how, about how to incorporate that in your research, please contact Eric or I about technology that we have. We have a terrestrial LiDAR, we have drone with multispectral and hyperspectral camera, and we have computers in the lab that have remote sensing software and GIS software.
Carla, Frank put a question in the chat section.
There's a question. At the end of your talk, you stated that there could be losses of pollinators in Northeast US. what about Illinois and Indiana and West and Middle Tennessee? The Midwest area is what caught his eye.
Yeah, as I was writing that slide, I thought it's not really, am I sharing the prop? No, this one. It's too broad of a generalization. Let's see if I can go back to that slide, but you're right, yes, there is Midwest, this one. Yeah, so I guess we focused more on areas that have proportionally, that are proportionally high in tomato crops. So focusing on the basically purple, because that's where you have a high proportion of the crops that are grown in that county. A high proportion of those crops is represented by tomatoes. But you're right. I mean, if we just map counties that have a loss, so purple is loss in buzz-pollinators for tomatoes. It's a pretty broad area, right?
Okay, that looks like those are all the questions for today. I want to thank Dr. Papeş for sharing with us today. And if you have any questions or in the future, please contact her or Dr. Greg Wiggins about collaborations. And so